refers to the segment of DNA deoxyribonucleic acid that contains the genetic instructions or code for the synthesis of a specific protein or functional RNA molecule. Meanwhile, clone refers to the collection of molecules or cells all identical to an original molecule or cells. Therefore, gene cloning, also known as DNA cloning, molecular cloning, or recombinant engineering, is a technique used to produce multiple and identical copies of a specific gene or a piece of DNA, in which they can produce either a wild-type gene, which is a normal copy of the gene, or mutant-type gene, which is the altered version of the gene. The principle of gene cloning involves a process where a fragment of DNA containing the desired gene is isolated from an organism. The isolated DNA segment is integrated into suitable vector, forming recombinant DNA. The recombinant DNA is transferred to the host cell, usually a bacteria, in which the recombinant DNA replicates as the bacterial cell divides, thus forming the copies of the recombinant DNA. After a large number of cell divisions, multiple copies or a clone of identical cells are produced. Each new cell in the colony contains one or more copies of the recombinant DNA molecule, where the gene of interest carried by the recombinant molecule is now said to be cloned. However, sometimes the host cells may need to use the genetic information in the recombinant DNA to make proteins.
The basic requirements for gene cloning include a source gene of interest which contains the desired genetic sequence or coding region, and it needs to be isolated from its source. The source of gene interest can be obtained from a variety of organisms such as bacteria, plants, or animals. Next, the restriction enzymes to cut DNA into fragments, and DNA ligases are then used to seal the DNA fragments together. Then, another basic requirement for gene cloning is a vector, whether cloning vector or expression vector, which is used to carry and replicate the gene of interest. The vector should have features such as a selectable marker and an origin of replication. Lastly, an host organism, which is the clone DNA, will be replicated and expressed.
The first step involved in gene cloning is isolation of the DNA fragment or gene of interest. At first, a donor DNA fragment should be isolated. There are two methods for isolation of the desired gene or DNA fragment. First, by using restriction endonuclease enzyme. The enzyme restriction endonuclease is a key enzyme in molecular gene cloning. It has specific restriction site for its action. The enzyme restriction endonuclease generates a DNA fragment either with blunt end or with sticky ends. Second, by using reverse transcriptase enzyme. Reverse transcriptase enzyme synthesizes complementary DNA strand of the desired gene using its mRNA. DNA fragment is cut with a restriction enzyme. Restriction enzyme is highly specific and will only cut DNA at specific base sequences, which is 4 to 8 base pairs long. Allen and H3 will produce blunt ends. BAM-H1, HIN3, and EcoR1 produce sticky ends. The second step involved is insertion of gene of interest into a vector. When donor DNA fragment is incorporated into a host cell, it will not replicate because the isolated gene do not have the capacity to replicate itself. Therefore, before introduction of donor fragment into host, a suitable vector should be selected. Vector is the self-replicating DNA molecules that are used to transfer foreign DNA fragment into host cell. Examples of vector are bacterial plasmids, bacteriophage, bacterial artificial chromosomes, yeast artificial chromosomes, and mammalian artificial chromosomes. However, the most common vectors are bacterial plasmids and bacteriophage. Insertion of DNA fragment into plasmid is when the plasmid vector is cut open by the same restriction endonuclease enzyme used for the isolation of donor DNA fragment. The combination of donor DNA fragment and plasmid vector are combined together. In the presence of DNA ligase, base pairing of the donor DNA fragment and plasmid vector forming recombinant vector. The characteristics of cloning vector include it must be self-replicating inside host cell, such as ORI. Second, it must possess a restriction site for restriction endonuclease enzymes, for example, EcoR1 and PIVOL, so that plasmid can be cut in one place only and insert the foreign gene to be cloned. Next, it must be circular so that it is less likely to be broken down. Besides, it must contain marker genes, which is antibiotic-resistant marker gene, AMP with superscript R is denoted for ampicillin resistant. And lastly, introduction of donor DNA fragment must not interfere with replication property of the vector. The cloning vector does not necessarily help to express a protein which the foreign DNA encodes. Therefore, expression vector is needed. An expression vector is a plasmid that not only introduces gene of interest into host organism, normally Escherichia coli, but also aids in the analysis of the gene of interest via relevant protein product expression. Only a plasmid can be expression vector. An expression vector consists of regulatory elements like enhancers, promoters, termination sequences, transcription initiation site, and translation initiation sites, in addition to having all the features of a typical cloning vector.
The third step is transfer vector into host cells. The process of transferring the recombinant vector into cell usually referred by 4 different terminologies, which are transformation, conjugation, electroporation, and microinjection. Transformation is the process of introducing free DNA into bacteria. It usually used to insert the recombinant DNA molecule into a competent E. coli cells. The cells must be made competent by treating with calcium chloride that facilitates binding of vector to the surface of E. coli cell, followed by heat shock at 42 degrees Celsius for 2 minutes to facilitate entry of vector into cells. Conjugation is the contact of bacteria that involve the exchange of genetic material. The process of conjugation started with donor cell attached to a recipient cell with its pilus. The pilus cause the cells to draw together. Then, the cells contact one another. After that, one strand of plasmid DNA is transferred to the recipient. The recipient synthesizes a complementary strand to become an F-positive cell, while the donor synthesizes a complementary strand restoring its complete plasmid. For electroporation, it uses an electrical pulse to create temporary pores on cell membranes through which substance like nucleic acid can pass into cells. Electroporation is a pretty easy thing to do. Host cells and certain molecules are put in a conductive fluid and surrounded by a closed electrical circuit. A short electrical pulse is sent through the cell suspension. It has the optimum voltage and only lasts a few microseconds to a millisecond. This upsets the membrane's phospholipid bilayer, which causes brief pores to form. At the same time, the electric potential across the cell membrane rises, which lets charged molecules like DNA move across the membrane through the pores, similar to how electrophoresis works. After electroporation, the cell membrane recovers and expression of the transfected nucleic acid can occur. Lastly, microinjection is a technique of delivering foreign DNA into a single living cell using a very thin needle loading pipette. It is a microsurgical process done on a single cell with a glass needle and a precision positioning device called a micromanipulator to control the movement of the micropipette and a microinjector. Hydrostatic pressure is used to push the fluid with the genetic material out of the micropipette. Most injections are done under direct vision control with the help of a microscope. Together, the small tip size of these micropipettes and the high precision of the micromanipulator make it possible to deliver DNA with accuracy and precision.
Well, the next step is multiplication of the host cells, where the transformed plasmid will be replicated at each bacterial cell division. This results in the production of many clones of gene of interest. Since transformation generates a mixed population with both recombinant and non-recombinant host cells. To multiply the recombinant cells or host cell that contain gene of interest by cellular replication, they need to be isolated from non-recombinant cell by utilizing the marker gene of plasmid. For example, LEC-operon, which is a group of gene with single promoter in E. coli, contain LEC-Z gene that codes for beta-galactosidase enzyme. This enzyme hydrolyzes lactose into glucose and galactose and hydrolyzes colorless X-gal into glucose and blue color pigment. A fully active beta-galactosidase enzyme contains alpha and beta fragment, which requires the expression of both LEC-Z alpha and LEC-Z beta genes. Therefore, for the selection to occur, the host cells are plated onto a medium containing ampicillin as a selection marker, IPTG to induce the expression of LEC-Z gene, and X-gal to detect the presence of beta-galactosidase. In non-recombinant cells, LEC-Z gene is expressed. Fully active beta-galactosidase is produced to hydrolyze X-gal to produce blue color colonies. In contrast, in recombinant cells, LEC-Z gene is replaced by the inserted gene of interest. Therefore, no beta-galactosidase is produced to hydrolyze X-gal, leading to production of white color colonies. After selecting and separating the recombinant cells, the cells are then allowed to grow and multiply to produce many clones of gene of interest under optimum conditions. Lastly, the product of interest like plasmid DNA or proteins are isolated and purified to extract them from bacterial culture. The steps include pelleting the bacterial culture by centrifugation and remove the supernatant, adding lysis buffer to lyse the bacteria by disrupting the bacterial membranes, adding neutralization buffer to neutralize the alkaline conditions and fix the plasmid DNA onto the silica filter, repeated washing steps with washing buffer, and lastly, elution of plasmid DNA with sterile water or an elution buffer by centrifugation. If the product of interest are proteins, they are usually fused to a protein tag for protein purification.
To know that the gene of interest has successfully cloned, some method can be used to screen for the positive result. First method used is by using blue-white screening method. It is a widely used method which relies on LATZ gene and codes the enzyme beta-galactosidase which can hydrolyze lactose. Blue-white screens rely on the vectors where the gene of interest is inserted into the middle of the LATZ gene, thus disrupting beta-galactosidase activity. Hence, if there is a presence of the gene of interest, cells cannot break down at galactose and turns white instead of blue, which indicate the gene of interest is successfully inserted into the plasmid. Next method to verify that the colonies containing plasmid with the gene of interest is by using restriction digest. In this method, restriction mapping should be performed to identify which restriction enzyme can be used to identify the presence of the insert within the plasmids. For example, the enzyme chosen can only cut once on either side of the expected insert. If the plasmid contains the gene of interest, two bands will appear in which one representing the vector backbone and the other representing the insert that indicate the gene of interest is successfully inserted within the plasmid. And lastly, colony polymerase chain reaction PCR can be used to detect the presence of or absence of DNA by using live bacterial colonies. In colony PCR, primers either that are specific to the insert or primers that are vector specific can be used. For example, if the primer used are specific to the insert, they will be positive for PCR product if the plasmid contain the gene of interest. Positive PCR product indicate the gene of interest had been successfully inserted into the plasmid.
Next, I will explain about the applications of gene cloning. Gene cloning have contributed in various fields such as in medicine and pharmaceutical industry, agriculture industry, and scientific research. In medicine and pharmaceutical industry, gene cloning is used in gene therapy to treat cancer such as leukemia and sickle cell anemia. The defective gene is removed and replaced by inserting a new and healthy gene. Other than that, gene cloning can be used for the production of various human proteins to treat human disease. Various vitamins, antibodies, and hormones can be produced rapidly. For example, insulin is used to treat diabetes. Human growth hormones is used to treat stunted growth. Blood clotting factors is used to treat hemophilia. Human albumin is used in treating hypoalbuminemia patients with severe injuries or infection. And hepatitis B vaccines is produced to protect humans from hepatitis B infection. In agricultural industry, genetically modified organisms or GMO crops are produced to meet specific requirements. GMO crops have better nutritional value, highly tolerant to herbicides, and have cheaper price than non-GMO crops. It is because GMO crops can be produced in a relatively short period of time, and these crops are less dependent on pesticides as they are genetically pest-resistant. For example, golden rice, which contain high vitamin A, and GMO corn, which can resist insect pesticide. In scientific research, gene cloning study can help researchers in understanding the function and regulation of various genes. By doing that, they can investigate its biological process, development, metabolism, and how it is associated with disease. Therefore, it can help in investigating the causes of various genetic disorders,
authors develop strategies to correct them.
As a conclusion, gene cloning is a technique used to create identical copies of specific DNA sequences. It can isolate and replicate genes of interest, facilitating the study of gene function and protein production. The process of gene cloning involves isolation of gene interest, inserting the gene interest into a vector, transfer vector into host cell, multiplication of host cell by cell division, and isolate and purify the desired product. In order to ensure the quality of cloning, several tests can be conducted, including blue-white screening, restriction digest, and colony polymerase chain reaction. These tests help ensure the reliability and effectiveness of gene cloning technique. And there are many products that come from gene cloning technique, such as we can develop new treatments and therapies in medicine and pharmaceutical industry. Next, develop herbicide tolerance in agriculture industry. And last but not least, it also helps in scientific research.